Hi, my name is Lorissa Saiz, and I work in the Kelleher lab here at UH, and we work on P elements in Drosophila melanogaster, or fruit flies. So we're interested specifically in P element transposition and finding means to repress or tolerate that transposition. So transposable elements are harmful, selfish genetic parasites that replicate in the germline of host genomes. There are two types of transposable elements, cut and paste and copy and paste. Transposable elements are extremely harmful, and they're extremely important to study because they make up about 50% of the human genome and about 85% of the corn or maize genome. One of the effects of P elements on fruit flies is hybrid dysgenesis. This occurs in offspring of parents in which the maternal strain is naive or does not contain P elements, and the P strain or paternal strain that does contain P elements. So in these F1 offspring, they can either be sterile, which means that their ovaries have atrophied and they are sensitive to P element transposition, or they can be normal, which means that they have developed some means of tolerance or resistance or silencing to P elements. So we did 3 different assays. Our first was the fertility assay, which we did with these hybrid dysgenic F1 offspring. So you can see from the chart here, in red we have these sensitive variants, and in the gray we have the tolerant variants. So our proportion of F1 atrophy was significantly lower in Bruno tolerant variations than in the sensitive variations across the board. This suggests that Bruno may have a role in tolerance or repression of P element transposition. We also did an expression assay in which we compared P element expression relative to the RPL32 control. The difference in expression was not statistically significant, again, across the board. You can see the tolerant strains are in red and the sensitive strains are in gray. So if P element copy number is not statistically significantly different than the RPL32 control across the board, then tolerance is the expected mechanism of P element silencing or P element control in Drosophila, or at least in these strains, the Bruno loss of function strains. We also did a P element excision assay in which we looked at the singed locus. So if you see our wild type flies here have no P elements present in their singed locus. Our singed weak flies have a pair of inverted P elements in their singed weak locus. So if we introduce a transpose source here, this red P element has the ability to excise from the locus and move to somewhere else in the genome, thus causing this singed extreme phenotype where the little bristles in the fly's back look more like they've been lit on fire and singed than the wild type bristles, which look normal. So in this study, we saw that the wild type excision rate was far higher than the Bruno loss of function excision rate. So this is surprising because if Bruno was acting as a tolerance factor, then the excision rates across the mutants and wild type should have been very similar or the same. So this is inconsistent with our hypothesis that Bruno is a tolerance factor. So in conclusion, during our P element expression assay, we saw that Bruno sensitive and tolerant pairs expression in these two were not statistically significantly different. So that suggested to us that Bruno was acting as a P elements tolerant factor. We found that P element excision rates were surprisingly way lower in the Bruno loss of function mutants than in the wild type, which was strange, but this suggests to us that Bruno may be acting as an upstream regulator of P element activity rather than conferring tolerance. Next, we need to do a P element expression assay on the Bruno loss of function dysgenic offspring that we did the excision assay on, and we need to do an excision assay on the Bruno tolerant and sensitive naturally occurring pairs. These are my references, and I want to say a big thank you to my PI and my lab mates who helped me along with this project and continue to help me as I finish. And thank you so much for listening to my talk.